Good afternoon to everyone. Today I'm going to present to you the results of my research on agar gels, developed in collaboration with the University of Torino, University of Milano Bicocca, the Institute of Conservation and Valorization of Cultural Heritage, and the Institute of Chemical Methodology of CNR. I would like to introduce the structure of my presentation, starting from a short introduction of gels in conservation, dealing with agar gels. I'm focused on an unsolved conservation problem, such as copper stain. Then I introduce you the goal of my research, focused on the study of different raw material, and in particular on the differences in chemical composition, the study of the water state, and the study also of the transport phenomena, and finally copper stain, the study of the removal of the copper from stone. Final conclusion and open problems. As many of you already know, since 40 years, gels are used in the field of conservation with great results because they satisfy the correct cleaning method, the requirement of a correct cleaning method, such as the selectivity, the effectivity, and the controllability, and so on. But the main advantages in using gel are the reduction of the transport phenomena and the control, the confine of water release close to the surface, with the reduction of the aggressiveness of the cleaning process. These are the main decay on which we can found on bronze metal, and also we can find also stain of corrosion product on marble faced with the bronze element. The typical patina, the typical corrosion patina are, in case of bronze element, are copper sulfate, such as anthracite, pozniakite, and brochantite. But we can also find chloride, like atacamite and paratacamite. These latter cases are the most dangerous product because active during the corrosion process. In my research, I'm going to focus on the removal of copper stain and in particular of brochantite. My research is developed with the remark of restorers. Some restorers noticed that applying agar gels, we have an extraordinary capacity in extraction of metal ion. To understand what is the cleaning mechanism, I want to introduce the chemical composition of agar, as already explained by Paolo Cremonesi. And agar gel is a natural polymer extracted from red seaweed. Its main components are 2-polysaccharide, agarose, and agaropectin. Agarose is a linear polymer consisting of alternated D-galactose and 3,6-anhydro-L-galactopyranose. Agaropectin is an heterogeneous agarose deeply modified with acidic side group, which reduces the gelling fraction and the gelling properties. I said that my research is developed with the restorers and is developed from a previous research in which an Italian provider wanted to test 10 different raw materials to identify the best product to be included in their catalog. And during this research, restorers noticed that different agar powders show different performances. These are the starting question of my research, such as are these differences so important, and how I have to choose the agar powder, and more, which is the real cleaning mechanism and to what extent this mechanism could influence the performances. To start, we have developed the research in 3 different steps. The first one is focused on the identification of chemical composition and differences. Then we study the water state and the transport phenomena to better understand the cleaning mechanism. And finally, we want to transfer this knowledge on a real case faced with the copper stain removal. To do that, we have chosen 4 different raw materials, 2 used in the field of conservation in Italy, of course, and the other 2 used with other application, Agar Sigma for biological application and Agar Food for food industry. The main theme of my research is a multi-analytical approach where each technique could provide specific informations. Let's move to the results. I carried out FTIR and FT-Raman spectroscopy, and as you could see, the FT-Raman and the FTIR spectra shows the typical absorption and scattering band of polysaccharide, but thanks to spectral subtraction, we found an extra feature in Agar Purissimo, one of the agar used in field conservation, and Agar Food, the agar used in food industry. These data are also confirmed by pyrolysis gas chromatography. We have carried out this analysis with a thermally assisted hydrolysis and methylation method. Thanks to this method, we are able to confirm the presence of galactose, but we can also support the extra feature that we have detected with FT-Raman spectra, in which we have found the presence of free glucose. And more, we have also found the presence of a typical compound of cellulose. So we can say that we have the presence, we can support the FT-Raman spectra with the presence of free glucose in BS and food, as you could see also in a quantitative analysis. And we can also say that agar gels used in the field of conservation has a different level of purification. An important step of my research, I said that, is the study of the water state. According to the literature, we know that water in hydrogels could be inbound and free form. So it is so interesting to understand at what extent the presence of bound and free water could influence the transport phenomena. To do that, we have modified the Italian standard unit for the study of the water absorption by capillarity, substituting the multi-filter papers with agar gels at different concentration. As you could see from the graph from the table, increasing the concentration of agarose of agar gel, we can find a decrease of the absorption capillarity coefficient, and moreover, we noticed that the agar at 1% weight on volume exhibits a similar trend of that we can record with a multi-filter paper. So we can say that the agar gels and 1% weight on volume, it's like a source of free water. The water state is also a study with unilateral NMR. According to the literature, we know that the major relaxation phenomena are longitudinal and transverse relaxation time. And we know that if I have only water, T1 and T2, the longitudinal and the transverse relaxation time, are of the order of a second and show a non-exponential trend. If I have a macromolecular like agarose or agar in this case, T2 is of the order of millisecond and show a exponential trend. This is we can say that the presence of agarose influenced the transport phenomena and the movement of the free water. This is confirmed by the longitudinal relaxation rate versus the concentration, and we can see the linear tendency with the increase of concentration. So increasing the concentration, water mobility decrease. In order to understand what is exactly T2, the transverse relaxation rate, 2 proton species must be taken into account. sorry. One is related to the bound water and the other one to the free water. The bound water has a short transverse relaxation time and the free water a long transverse relaxation time. As you could see from the graph, we have, when we increase the agar concentration, the transverse relaxation rate increase. And the slope of T2B is steeper than the slope of T2A. So we can say that increasing the concentration, water mobility decrease. Because we have a densification of the structure and water can move as it wants. We have also, in correlation with the study of the transport phenomena as well, we have tested the thickness of the gel, and always with NMR. And we noticed that we have tested the thickness of Agar Art, the agar used in the field of conservation, and Agar Sigma, the agar used for biological application. And what we have noticed that after 1 hour of contact, we have a decrease, a reduction of 50% of the volume. But also we have probably a deformation of the structure. Moreover, we have also noticed that the agar used in the field of conservation controls better the release, so it is more suitable for conservation works. For this reason, we have applied it for the real case, and I have said that I focus my research on copper stain, but to do that, to face this problem, I need a model sample. Two different approaches were followed. In the first attempt, marble specimens were stained with a solution of a copper sulfate solution by capillarity for 72 hours. In the second attempt, marble specimens were stained with a solution of copper sulfate and sodium carbonate released by dripping for 1 hour, by immersion in this case. Which are the differences? Firstly, the color, more intense in the second attempt and quite flammable in the other one. But secondly, the composition of the cells that we have synthesized. In the first case, X-ray diffraction detect the presence of Pozniakite. In the second attempt, Pozniakite and brochantite. Notwithstanding these results, we have used the first attempt because we are faced on this kind of real case. In this, we have a copper stain on the base of Napoleon statue sculpted by Antonio Canova, located in the courtyard of Brera Academy. We have tested 6 different formulations, pure agar gels and additivated agar gels. For the pure agar gels, we have used 3 different concentrations, 1, 3, and 5% weight on volume. For the other gels, we have used different additives with the same concentration. 3% weight on volume for agar gels and 1% for the additives. The additives were ADTA alkalified with ammonium carbonate, and triammonium citrate. Here you can see the Ecp analysis, the results of Ecp analysis for the evaluation of copper amount. And as you could see from the histogram, we found that for pure agar gels, after 1 hour of contact, the copper amount is lower than 50 microgram. On the other hand, with additivated agar gels, we have good results, and the copper amount exceeds quite in double. In the real case, we have a different trend. For about pure agar gels, after 1 hour of contact, the copper amount is dramatically dropped. On the contrary, for additivated agar gels, the trend is quite similar to that obtained in model samples. In the case of Napoleon Statue, we have used the best formulation, such as in which we have found the best results, such as Agar Art at 3% with triammonium citrate. In conclusion, we can say that thanks to vibrational spectroscopy and pyrolysis gas chromatography, we are able to detect differences in composition, and in particular we have found the presence of free glucose and cellulose derivatives, and we can say that we have a different level of purification that could influence the cleaning performances. Moreover, about the NMR analysis, we can say that we have a strictly dependency between water and concentration too. And in particular, we found that Agar Art, the agar used in the field of conservation, it controls better and it is more suitable for the restoration works. About the copper stain, we have optimized the deposition of brochantite directly on the model sample, and about the cleaning performances, as expected, we noticed that additivated agar gels works better compared to the pure one, in spite of an interesting capacity of complexation also of the pure agar gels. Open problems. My research is continue in optimizing the cleaning performances, and in particular focusing on the study of kinetic behavior of copper complex.